So just quickly, the TCA cycle is responsible for generating over half of the ATP from the oxidation of fuels. This is primarily because the substrate for the TCA cycle, acetyl-CoA, is generated by the oxidation of fatty acids, glucose, amino acids, as well as ketone bodies. With each turn of the TCA cycle, there's a net production of 3 NADH, FADH2, 2 carbon dioxide molecules, and 1 GTP for every acetyl-CoA that enters. The regulation of the TCA cycle is summarized here. There are 2 key regulatory enzymes to be aware of, and those are isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. These enzymes are very sensitive to the ratio of ATP to ADP, as well as the ratio of NADH to NAD in the cell. So in the case of isocitrate dehydrogenase, it's activated by ADP, and both enzymes are activated by calcium, which would be indicative of exercising muscle. The primary inhibitor for both of these enzymes will be NADH. As NADH levels accumulate, this will inhibit the activity of these enzymes and therefore reduce flux through the TCA cycle. Also of note is that alpha-ketoglutarate requires the same cofactors as the pyruvate dehydrogenase complex. So the entire cycle is summarized here. As acetyl-CoA enters, it will condense with oxaloacetate to make citrate. Again, our regulatory enzymes are down here, isocitrate dehydrogenase, which converts isocitrate to oxalosuccinate, and then oxalosuccinate to alpha-ketoglutarate through this 2-step reaction, and the conversion of alpha-ketoglutarate to succinyl-CoA by the alpha-ketoglutarate dehydrogenase complex. In these 2 regulatory reactions, you'll notice that decarboxylation occurs, and we produce NADH. Also of note are these biological inhibitors of the TCA cycle: fluoroacetate, which will inhibit the enzyme aconidase; arsenate, which will inhibit the alpha-ketoglutarate dehydrogenase complex; and malonate, which will inhibit succinate dehydrogenase. The other thing to remember here is that succinate dehydrogenase is also complex 2 in the electron transport chain. So it's important to note that in addition to the oxidation of acetyl-CoA through the TCA cycle, as we typically think about the TCA cycle, there's important contributions by the TCA cycle to other metabolic pathways. And those are largely summarized in this figure. And what you can see just globally is that there's lots of intermediates of the TCA cycle that come out of the cycle to be used as a substrate for other processes, as well as other substrates such as amino acids which enter into the TCA cycle to contribute to the carbon within the cycle itself. So some of the key intermediates to be aware of are shown here. First of all, oxaloacetate. This can be directly transaminated to aspartate or vice versa. Therefore, this amino acid can be directly transaminated to generate a TCA cycle intermediate. The same is true for alpha-ketoglutarate, which can be transaminated to glutamate. So glutamate, through the process of transamination, can directly contribute alpha-ketoglutarate to the cycle. The other amino acid contributors of the cycle are shown here. So generally, amino acids are a large contributor to replenish the intermediates of the TCA cycle through anaplerotic reactions. The other contributor, propionate, is a product of odd-chain fatty acid oxidation, and this will ultimately enter the cycle as succinyl-CoA. Alanine, transaminated to generate pyruvate, will also replenish the TCA cycle through the pyruvate carboxylase reaction. So there's 2 major compounds that are then taken out of the TCA cycle that we'll talk about. The first is oxaloacetate. So oxaloacetate can be removed from the TCA cycle in a process for gluconeogenesis to generate glucose in the fasted state. In that case, the reduced form of oxaloacetate or malate is shuttled out of the TCA cycle into the cytosol, where it can be reconverted to phosphoenol pyruvate through a series of reactions which involves the enzyme phosphoenol pyruvate carboxykinase. We'll address this in a couple weeks. The other major contributor to external pathways is citrate, which can be shuttled out of the TCA cycle and used in the process of fatty acid synthesis. So the removal of citrate from the mitochondria into the cytosol for fatty acid synthesis is shown here, where again, this is primarily in the fed state where excess glucose oxidation increases acetyl-CoA, and this is used to produce citrate, which will be shuttled out and used for fatty acid synthesis. Through a series of reactions we'll see in a few weeks, this citrate is cleaved by citrate lyase to generate acetyl-CoA, our substrate for fatty acid synthesis, and oxaloacetate. This oxaloacetate, through a two-step reaction involving malate dehydrogenase and malic enzyme, is reconverted back to pyruvate, which can enter back into the mitochondria and be reconverted to oxaloacetate through the enzyme pyruvate carboxylase. So let's finish with this question. So alcohol metabolism, which we haven't discussed, increases the production of NADH, increasing the ratio of NADH to NAD+ within the cell. So as a result of this, how would you expect increased consumption of alcohol to impact the activity of the TCA cycle? So to answer this question, all we really need to know is how is the TCA cycle regulated? Again, it's regulated at two key points: isocitrate dehydrogenase and alpha-ketoglutarate complex. We know that both of these enzymes are inhibited by high levels of NADH. Therefore, in the clinical condition when NADH is elevated, you would expect decreased activity of the TCA cycle.